Hey guys, Mrs. Horn here. Today it's the muscular system. Last chapter we learned about how messages are sent in the body and how an impulse will travel through a neuron. Well, those impulses are usually sending messages to muscles. So we're going to start off with the skeletal muscle. We're going to zoom in and zoom in and zoom in and see what a skeletal muscle is made up of, right down to the very cell. Then we'll talk about how muscles contract. I have a lot of diagrams that need to be labeled, and we'll talk about fast and slow muscle fibers, fibers that make up the muscles. So let's get started. This is actually chapter 50 in the book, so if you see somewhere where it mentions a figure and it says chapter 49, that is not correct. So please ignore that first bullet. Skeletal muscles consist of very long fibers that run through the length of the muscle. Now, during the nervous system, I said that the shape of a cell determines its function. So the shape of a nerve cell determines its function, just like the shape of a muscle cell determines its function. Muscle cells are very, very long, and they run the whole length of a muscle. So think of a quad muscle or your bicep or your tricep muscle, and the cells in there are the entire length of that muscle. Each fiber is one cell. Each fiber has more than one nuclei. So this type of cell is unique in that it has many nuclei in one cell. I'm going to get to a picture of all of this, so let's get all of the notes, and then I'll show you in the picture what and where everything is. Not only does each fiber have more than one nuclei, but each fiber consists of something called a myofibril. The myofibrils consist of two different kinds of myofilaments. There are thin and there are thick. The thin myofilaments are made up of a substance called actin. The thick are made up of myosin. The myofilaments have a pattern. It's with light and dark bands. We call those bands sarcomeres. The sarcomeres have borders in between them, and those are going to be labeled Z-lines on the diagram I'm about to show you. There are also structures labeled A band or an I band. Now the A band is going to be the region that's the length of that thick filament. The I band is going to be the region of only thin filaments. That's at the very edge of the sarcomere. So on page 38 of your packet is a diagram that maps out all of these details. Mine is already labeled, so you have to add some labels to yours. So if we start with the bicep and we look at this muscle, let's pull out just one bundle of fibers. So here's one bundle. Now that bundle is made up of many fibers. So we've got 9 fibers that are making up the bundle. If we pull out just one fiber, now that one fiber is one cell. This one cell has more than one nuclei in it. If we look even further and look at what's in that cell, the outside of it is a plasma membrane, which is the same structure of a plasma membrane as in your the nerve cells or skin cells or liver cells. It's the same bilipid layer. Inside the plasma membrane, things start to look a little bit different. Here's where the structure of this cell is very unique. Inside of this one cell, we have many myofibrils. There's a lot in there. It looks like a bunch of spaghetti in there. If we pull out one myofibril, we'll see that there are some bands on it. There are some areas that look very light, and then there are some areas that look a little darker. That's what's going to make up this banding pattern. So we have a dark band, we have some light bands. This line that runs right through the middle of the light band, that's called a Z-line. If you go from line to line, that distance is a sarcomere. The other bands that I talked about. This is the band that's the region of the thick filaments. This is the A band. It's not actually labeled on here, but from here to here is the A band, and the I band is where there's nothing right now. It's just the thin. It's just the yellow. So it looks very open. It actually makes it looks kind of light. That's going to be your I band. The area of the sarcomere that looks very dark, that's going to be your A band. Also, that's labeled on here, the purple are the myosin. Those are the thick filaments, and they look fatter. They look thicker than the other ones. And the actin are going to be the thin, narrow, yellow strands of filaments. In order for a muscle to contract, that sarcomere is going to shrink. So that distance from Z-band to the Z-line to Z-line, that's going to shrink and everything is going to push into the center. The Z-line will become shorter. The A-band, remember the A-band was the length of the thick filament, that's not going to change. The A-band is going to stay the same. The I-band, which that was the band of the light color band, that's going to shorten. It's actually might even look like it doesn't even exist. The H band, which is in the middle, that's going to completely disappear. It's not even going to exist when the muscle contracts. This whole muscle contraction is known as the sliding filament model. This is where filaments are going to stay the same size. So the myosin and the actin never change length or shape or size. They're always going to stay the same, but they are simply sliding. They're sliding past each other. And when it slides in, that's a contraction. When it slides back, that's when the muscle gets to relax. As those filaments are going to overlap and the overlapping starts to increase, that I band and the H zone, they're going to look like they're completely eliminated. And the sliding is based on the interaction between the actin of the thin and the myosin of the thick. And I've got some more details on that in a few pages. This is not the picture that you have on page 40, but mine is similar. I do have some things labeled on here. My diagram is more condensed than yours, but I wanted to show it to you because this is where the muscle is very relaxed, and the size of the sarcomere is the biggest. From Z-band to Z-band, it looks pretty wide. As the muscle begins to contract, those Z-lines are coming closer and closer, and then when the muscle is fully contracted, those Z-lines are the closest. The filaments are the most overlapped. There's no I-band anymore. See this right here where it's nice and light? That's the I-band. But when it's contracted, the I-band, you can't even see it because everything is so overlapped. And then these pictures here, these are pictures from a scanning electron microscope of what these muscle fibers look like when they contract. And when it's relaxed, you can definitely see a light band, dark band, light band. And then as it contracts, it all looks dark. There's no open space. So I said before that the sliding filament model, it has to do with interactions between the myosin and the actin. So here's the diagram that would explain how those two filaments interact. Please start with labeling all the pictures. So we have 1 through 5. And this is a view of both the thick filament, which is the myosin, and the thin, which is the actin. So I've got 2 actin layers with the thick, the myosin in the middle. And then we're zooming in. We're going to zoom in nice and close to get some close-up of that. This process uses a lot of ATP. So this is something that happens in your skeletal muscles, so voluntary muscles. We're going to use a lot of ATP here in order for this to happen. The purple, the thick filament, has what's called a myosin head on it. And if we look in the next step, what's happening is that the energy was used. It was used to move the myosin head to a different configuration. And once it's in that spot, it's going to attach. It's going to attach to a myosin binding site. On my picture, it's a little more noticeable than yours, but if you look at these, they look like olives. And if you look at where the opening of the olive would be, that is a myosin binding site. So it looks a little darker shaded on your picture maybe. But this myosin head has to bind to one of those myosin binding sites, which is part of the actin. And once it does that, it is going to move it. It's going to bind, and it's going to push it. And it's pushing that thin filament towards the center of the sarcomere, and that's what's allowing it to contract. And then after it does that, it's going to reset. And now we need another molecule of ATP to do the whole thing over again. So where it shows ADP and then the P, that's, remember ADP, it's adenosine diphosphate. It's a low energy version of ATP. But ATP is broken up into the ADP and the phosphate. So after you use ATP, those are the two molecules that are the end result. So quickly, using ATP, reconfiguring that myosin head to getting it into kind of a start position, it's going to look for a myosin binding site, it's going to attach to it, and it's going to fling that thin filament towards the center of the sarcomere, which is going to enable the muscle to contract. And then it's going to reset, and then we need another molecule of ATP to continue on. This is not just one of these things is happening to contract a muscle. There's hundreds, thousands of these things happening all at once to contract just one of your skeletal muscles. Controlling a muscle contraction. So when the muscle is relaxed, those myosin binding sites are blocked by something called tropomyosin, which is a protein. There's a picture at the bottom, and I'll show you what that looks like in just a minute. But when the muscle is relaxed, those binding sites, they don't want the myosin to bind to them, so it covers up the binding sites. If the binding site was open all the time, then your muscles would be contracted all the time, and you probably would move around a little funny. When the muscle contracts, those myosin binding sites are now uncovered by calcium ions. So here's where having calcium ions in your body is a really important thing to have in your diet. The calcium concentration is regulated by a new organelle we haven't talked about before. It's called the sarcoplasmic reticulum, and it's a kind of similar to the endoplasmic reticulum, but its only job is to regulate calcium concentration. All right, so here's the picture. Similar to what we just had, we have the actin, which look like those bunch of olives. What I haven't shown you yet is this string, maybe a piece of string or a piece of spaghetti that's going to look like it's wrapping around in a spiral staircase around, it's tropomyosin. This is a protein. And on the tropomyosin, we have some calcium binding sites. Now, in this picture, the myosin binding sites are blocked, which means up here, the muscle is relaxed. So it's that tropomyosin, if you look, is covering all of those olive openings. It's covering all those sites. So nothing, the muscle cannot contract right now. However, if calcium ions are in the area and they are available, the calcium is going to connect to the complex, and it's going to move that tropomyosin out of the way. And now the myosin binding sites are exposed, those little olive openings, and now the myosin head is available to connect to the actin and slide it towards the center of the sarcomere. So muscle relaxed, all the binding sites are covered, calcium is available, calcium will move the tropomyosin and move it out of the way, so now the myosin head can attach and the muscle can contract. The last thing I want to talk about are fast and slow muscle fibers. Now, how long can your muscle contract will depend on how long that calcium concentration level will remain high in your cells. And I put cytosol here because that's the cytoplasm area of a cell. So how long can you hold a muscle contraction depends on how much calcium is available in that space. There are two different types of muscle fibers based on the duration of their twitches. A fast muscle fiber is used for very short, very rapid, powerful contractions. So this might be sprinting, could even be standing up quick, jumping, lifting weights. This would be a fast muscle fibers are used for that. Slow muscle fibers are used for long, sustaining contractions. Standing up for a long period of time, even sitting in your seat and sitting up properly for a long period of time. Those are going to be slow muscle fibers. A muscle fiber that has fewer sarcoplasmic reticulum has a slower calcium pump than a fast fiber. So the calcium is going to hang out in the cytosol for a little bit longer, which allows that contraction to last longer. A twitch in a slow fiber can last 5 times as long as it would in a fast fiber because of all that extra calcium. And then the slow fibers use a steady supply of energy. They have a lot of mitochondria in every cell, and they have a protein called myoglobin, which is a protein that helps to store oxygen. So if they have a constant storage of oxygen, those cells can use the oxygen to carry on cellular respiration and make ATP, make a lot of ATP. And so your muscle cells have a lot more mitochondria than any other cell in your body. So that wraps up the muscular system, and I hope this helps.